very beautiful, yet they are very powerful also. They are beneficial also, and they are very dangerous also. Now we have an example of COVID-19, coronavirus, which has brought the whole world to its knees. So this is the power of microorganisms. Next, all life in the biosphere depend on microorganisms. If I say that we owe all the beings on the face of Earth, they owe their existence to microorganisms. Because had there been no microorganisms, then the entire world would have been turned into a heap of garbage. See the role played by the bacteria. They decompose all the waste matter. Otherwise, there would be so much accumulation of waste matter that there will be no life on Earth. So this is the importance of microorganisms. Next. So this is just to tell you the role played by the microorganisms. 140 million tons of nitrogen gas are removed from the atmosphere by microorganisms. More than 40% of the CO2 drawn from the atmosphere is accomplished by microorganisms. These are the services that they are performing, yet we cannot see them. So nobody knows them. So they are not given as much importance as other natural resources, say plants, are given. Marine microorganisms are responsible for producing and degrading dimethyl sulfide, a major climate cooling gas. So this is just a few examples of the ecosystem services performed by the microorganisms. Next. Now culture collections. Next. Microorganisms, just as I said that we have a national bureau of plant genetic resources where all the seeds of different varieties, different plants, cereals, fruits, vegetables, oil seeds, they are preserved. In the same way, their microorganisms are also equally important, so they also have to be protected and preserved. So the first culture collection was established in 1890 at German University of Prague. And in 1970, another very important body came into being, which is World Federation for Culture Collection, which is managing the culture collections all over the world. Next. Next. Now, there are total 712 culture collections in the world, in different countries. And out of these, in 73 countries. And this is the 2.5 million microbial resources are preserved. 2.5 million microbial resources are preserved in these culture collections. This is very minuscule, 2.5 million. It is very minuscule. And this is the number of bacteria and fungi which is being preserved in culture collection. This is not the latest figure, it has slightly changed. But this is just to give you an idea, only 2.5 million. So this is a very, very small fraction of the number of microorganisms that are there on the face of the earth which have been preserved. Next. Next. Now, we use the term microbial resource. What do we mean by microbial resource? Microbial strain, they should have certain qualities. They should be taxonomically defined, physiologically characterized, quality controlled, and also well documented. Then we use the term microbial resource. Because any microorganism, we cannot say it is a microbial resource. This is the exact definition of the term microbial resource. Next. Now look at this figure. A trillion species of microorganisms are supposed to exist in the biosphere. However, only 5% of them are described. In the case of bacteria, we say hardly 1% of the bacteria have been described, which means 99% of the bacterial species are unknown. In the case of fungi, at the most it is 5%, which means 95% of the microbial wealth is not known to us. It still remains to be discovered. Next. Now the culture collection. There are two types of culture collection. One is conventional culture collection. What they do is that they maintain the cultures and they distribute the cultures. They give the cultures to different stakeholders. But when we say microbial resource center, besides this, they perform other functions also. They have an expert knowledge. They provide the identification services. They intellectual property rights also. When I say bureau and institute, bureau means office, but bureau has slightly different function. They have certain legal aspects are involved in the tasks that they perform. In fact, our culture collection is a microbial resource center because we are here, we are preserving also, we are distributing also, we are giving the identification services also, and we deal with intellectual property matters also. Next. Now, next, this is just Now the top 10 strain holders of the world: USA, Japan, India comes at third one, and the 10th one is Belgium. This is the on the basis of the number of cultures which are being preserved in different cultural collections. India is at, that is why at third number we say India is one of the hotspot of biodiversity. Hotspot of biodiversity: Western Ghats and Eastern Himalayas. There are 12 mega biodiverse countries in the world, and in these 12 countries there are 25 biodiversity hotspots. And out of these, two biodiversity hotspots are India, Western Ghats and Eastern Himalayas. That is why we say that these have to be preserved. Our wealth lies in these regions, be it plant wealth or be it microbial wealth. Next. Now in India there are 30 culture collections. Now I was saying that 712 culture collections. Now we have 30 culture collections, and this is the number of strains which are being preserved in these culture collections, and number of patent deposits. I will be telling you what is it. This number of those culture collections is 3. Okay. Next. Now this is just a list of the culture collections. List of the culture collections in India. Next. Now this is, I was saying that from patent point of view. Now, as you know that under the Indian Patent Act, microorganisms cannot be patented. Microorganism per se cannot be patented. Seeds cannot be patented. Okay, lot of you must when negotiation and at the global level takes place, one of the point of contention is patent law of the country. It has been amended. Microbes cannot be patented. Okay, but microbial process can be patented. Say for example, bacteria is there, fungi are there, penicillin antibiotic. Okay, it is obtained from a penicillin. Okay, penicillin in India, for example, you cannot patent. So if you develop a process, so that process can be patented. Now, in order to patent that process, the culture of that microorganism has to be deposited in a culture collection which has the status of International Depository Authority. Now, in earlier what used to happen, say for example, if someone has developed a process of isolating an antibiotic, say from particular fungus or bacteria, you apply for a patent in India. Now you want to apply for a patent in 10 other countries also. So the earlier the requirement was that culture has to be deposited in 10 different countries. So you are dealing with a microorganism. It is very difficult to handle these microorganisms. Okay, and cumbersome also and expensive also. So in 1977, a treaty was enacted, which is referred to as the Budapest Treaty. India is signatory to this treaty, and this treaty came into force in 1980. Now under this treaty, इसको बंद कर दो, बंद कर दो, सभी लोग अपने मोबाइल साइलेंट मोड पे करो. Under this treaty, India is signatory. So what is the advantage of this treaty? Under this treaty, certain culture collections have been given the status of International Depository Authority. Now, if you are applying for a patent in India and you are depositing that culture, say for example in Mau, okay, then you can apply in 10 other countries for patent. Only requirement is that you have deposited the microorganism here. You don't need to deposit that culture in 10 different countries where you are applying for a patent. Now in India, this is being administered by World Intellectual Property Organization, which is based in Geneva. In India, there are 3 culture collections have been given the status of International Depository Authority. One is at Chandigarh, Microbial Type Culture Collection and Gene Bank, which deals with industrial microorganisms. These are agriculturally important microorganisms. They are maintaining industrially important microorganisms. Another is Microbial Culture Collection at Pune. And third one is in 2020, 2 years back, National Agricultural Microbial Culture Collection was given the status of International Depository Authority. Now we are receiving the cultures from multinational companies which are based in India. They are sending the cultures and we are depositing. Now this is Budapest, this is what I just wanted to tell you there. And here the requirement is, many people will not be knowing, once a microbe has been deposited, that microorganism has to be maintained for 30 years. That culture, it becomes the responsibility of the culture collection to maintain that culture for a period of 30 years. Confidentiality also has to be maintained, and not that anybody will be knowing that these are the cultures, because companies have deposited those. So a lot of pressures will also be there. Next, this is the I have next. Now there are in 24 countries, there are total number of IDAs 44. Maybe the number has increased by one or two. And this is the list of, I was told you that these are the culture collections which have been given the status of International Depository Authority. Next. It is a very big American type culture collection. It was the first culture collection which was given the status of IDA in 1981. It is a very big culture collection. Okay. Next. I'm telling you the list of big IDAs. DSMZ in Germany. Next. CABI, Commonwealth Agricultural Bureau. It is at UK. Next. China Center for Type Culture Collection. Next. MTCC. I was based at Chandigarh. Next. Microbial Culture Collection, again having the status of IDA. Next. 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 These specific culture collections are also there. For example, one the International Culture Collection, Arboricultural Mycorrhizal Fungi, Industrial Yeast Collection, Marine Culture Collection of India. They are preserving specific type of microorganisms. Next. 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 Now, this bureau was established in 2001 at New Delhi, and 2004 it was transferred to this place at Mount. Okay. Next. Now we are having this bureau is it is the culture collection affiliate member of World Federation for Culture Collections. We have a microbial genomic resource repository also where we are keeping the genetic material of microorganisms. We have a repository of that also. Then we have got high-end, well-equipped laboratories dealing with bioinformatics, dealing with genomics, dealing with preservation of microorganisms. Next. And another, another very important thing that I want to tell you is that we talk about biofertilizers, biopesticides, and so far as the biopesticide are concerned, nobody knew what is being sold in the market. The government was also not knowing. Nobody knew. Okay, so what they, because and a lot of spurious products were available in the market. Now the government is tightening the screws. Now the government took the decision 3 years back that any company which has to register a biopesticide with Central Insecticide Board and Registration Committee, they have made it mandatory that they will send that product to MAU. Here they will do the DNA fingerprinting of that product, which is being looked after by Dr. Alok Srivastava. He is doing it. That will be DNA fingerprinted. The report will be generated. That report will be sent to the Central Insecticide Board. Then and only then they will be issuing the certificate of registration. And earlier also the biopesticides which have been registered, they have also been asked to send the samples to this place. At least good beginning has been made, and we are having the database. We know what is being sold in the market. And spurious products can be caught because those the formulations which are registered, they have already been DNA fingerprinted. Next. 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 Now these are the objectives of the bureau. So what we do is identification. We say that we are a microbial resource center. Identification, characterization, documentation, studies on microbial diversity and systematics, conservation, maintenance, and utilization of microorganisms. And at the same time, the scientists, they are visiting the different ecological niches, different regions, collecting the samples from there, isolating the microorganisms, studying them, and preserving them in the culture collection. Next. Now this number has reached about 7,000. It is near about 7,000. And now bacteria, actinomyces, cyanobacteria, and fungi are being preserved. Now, these have been deposited by people, by researchers from all states from India, from Tamil Nadu to Kashmir, from Gujarat to Nagaland and Mizoram. People are the microbiologists, plant pathologists who are carrying out work on microorganisms. They are depositing the cultures here. We have the catalogs which are online, and the record of each and every microorganism is being maintained in those catalogs. Next, this is just to tell you the states bacterial collection. Next, actinomycetes. Next, this is just to give you so much diversity, so much variations on the basis of morphology, on the basis of color, etc. This is a hello archaea, Bacillus megaterium, Bacillus rhizobium physioli, all these bacteria, these bacterial isolates have been Car Nicobar Island. Next. Now this here we have KB UK 1427. Now what at the outset I said that they are not giving as much importance, because something which you can see with your own eyes, that is given more importance. Something which you cannot see with your own eyes, that is not so much important if you do not know. Now our country woke up to the importance of microorganisms very late. This bureau was established in 2001. Now can you imagine, earlier also, in the pre-independent times, in 1947, in 1950, at that time also the people were plant pathologists, microbiologists, they were studying microorganisms. But at that time, we were under British rule. Expertise for identification of microorganisms was not available in the country. So the people were sending those cultures to UK. International Mycological Institute is there. Why they were sending? Because the identification was free of charge. Now, at that time, 1940, 1944, 1950, the scientists had sent those cultures to UK, and they were preserving at Cabby. They had preserved it, these cultures. Now, when this bureau was established at that time, the people thought that our microbial wealth is lying with other countries. This is a wealth, microbial wealth, which cannot be uh, estimated, evaluated in terms of money. At that time, then it was negotiation at the government level. Earlier, they were reluctant to give them back. Then negotiation at the highest level, at the government level, took place, and then 1427 cultures were repatriated. They were maintained in life form. Here, those cultures were revived, and we are preserving those cultures. Now, we have our cultures in Germany also, and America also, but they are not going to return you, even if you pay them in pounds or in dollars. They are not going to return those cultures. Next, this is just the genera of fungi which are available in the culture collection. And this is a picture of the cultures which were in UK and which were repatriated from UK: Fusarium, Colatotrichum, Echinoderm, Gliomastrix. Next, all these cultures were repatriated. Some were sent in 1940, some 1944, some 1950, 1953. Next, then we have a mushroom cultures also in our culture collection. Next, this is just to give you a view of the. These are agricultural biocontrol agents, Trichoderma species, Bacillus species. They are available in our culture collection. Next. Now this is plant pathogenic, and we have beneficial microorganisms also. For example, used as a biofertilizer, biocontrol. At the same time, uh, the pathogens which are causing disease in different plants, those cultures are also, because they are also equally important. Each microbe has its own importance. Whether from the culture collection point of view, each microorganism has equal importance. Pathogenic microorganisms also as important as a beneficial microorganism, so far as its importance, its preservation is concerned. Next, next, next. entomopathogenic also. There are fungi which are used for controlling insects also, for Vivaria, meta-rhizium. There a lot of demand is there. A lot of demand of these cultures is there. Next, cyanobacterial culture collection is also there. We have a small collection of cyanobacteria also. Next, next, next. We will link this with different. This is a cyanobacterial culture collection is that in Farland they have also sent their cultures. We are maintaining those cultures also here. Next, next. Now, where from we have got these. We have these microorganisms have isolated from soil, rhizosphere soil, different ecological niches: water, hot water spring, hot desert, saline environment, cold deserts, cold springs, different ecological niches. Microorganisms have been isolated from different ecological niches. That's now. This is just to tell you. We went to me and along with one of the scientists who will be giving a talk. We went to Manipur Jhum area is there. We collected the samples from those. From Meghalaya also the scientists have visited and collected. From Salt Pan also the scientists have visited, and they are working on different projects. So as a part of those projects, they have collected the samples, and after carrying out the studies, they deposit the cultures in culture collection. Next. Now we have got linkages with different institutes also. Next. The pressure. Now this is very important. How we are maintaining the cultures in our culture collection. Short-term preservation is there. Long-term preservation is there. Now in short term, as you know, we are doing the subculturing. Subculturing you can maintain it for at 4 degrees, 4 to 6 for 4 to 6 weeks you can keep. Then we are maintaining. We will show you glycerol stock also in -80. We are preserving them glycerol and storing in -80. Then fungi we are storing in, preserving in mineral oil, parafilm oil. It is also a short-term preservation at the most 1 year. After that, again you have to revive it, and again you have to store it. I think this is the more lyophilization, freeze drying of cultures. This is the most important part of the bureau. This room where all the cultures are being maintained is in long-term preservation. Long-term, all the cultures that you see in Petri plate, they are brought to powdered form. The moisture content, the moisture is removed. There is a machine called lyophilizer. Moisture content is brought down to 2 to 3%. Cryoprotectants are also added, and this is the whole process will take 24 hours, and that machine will run for 22 hours continuously. There should not be a shutdown. Then these are preserved. This is long-term preservation. Another is liquid nitrogen also, -190. This is just picture. We are not maintaining the preservation of cultures in -180 or 190 degrees C in liquid nitrogen. And next. Now we had a facility, small facility at New Delhi also. We have kept some cultures. Next. Now what we are doing is, anybody can deposit the cultures in culture collection. There is a passport data form. In the open deposit category means you can deposit it. We will maintain, we will preserve those cultures. It will be free of charge. Okay, but it will be available to other researchers for research. Okay, for commercial purpose also there are rules for that. And then we are continuously supplying the cultures to different stakeholders, to colleges, to universities, to industry. Rates are different for those. So, and at the CIBIC, I told you. Then there's another category is there. For example, if someone has got a very good microorganism, very good culture of say Bacillus, they do not have the facility to maintain that culture. So they can deposit it here under safe deposit category. We will charge 5,000 per year from public sector and 8,000 per year from private sector. We will keep it safe. We will maintain it. We will take the charges. We will not give it to anybody. This is also, then registration also very important. Now all the microorganisms cannot be patented because it is not, but they may have other very useful traits. So another is that we have another service that we are registering the microorganisms which have some useful traits, good traits, or say bad traits, highly pathogenic, say for example. So what we are doing is we have got a committee. We receive the culture, we receive the form, then there is exports are there from outside. They are evaluating, though they are giving the recommendation whether this microorganism is fit enough to be registered or not. And accordingly, we are registering it for 10 years. It is again free of charge. This is also it is soft protection. Next, identification services we are giving. Identification services again, morphological identification of fungi and bacteria, molecular identification of fungi and bacteria. This is just the procedure that we are following. How we are, when you will be depositing general deposit, once we receive the culture along with the passport form, what we will do is we will check its viability and purity. And if it is, then after that we will send you a form. We will ask you and the culture also. Then we will ask you whether it is the same culture that you have deposited or it is a different culture. Because tomorrow you may say it is not the same culture that I have deposited. So you will say yes, it is the same culture that I have deposited. Then after that we will give you accession number and that it will go in the catalog. For industrial culture supply also, in culture supply we are taking a certificate, material transfer agreement, whether it is for research purpose only or commercial purpose also. Because if it is for commercial purpose, the rate will be different. And commercial purpose means, now this is very important. Earlier, many drugs which were isolated because patent regime. It is not in 50s and 60s there was no patent regime. So anybody, if someone cancer drug, I'm forgetting the name of the cancer drug. One of the scientists from Sweden went along with his family to an African country. There he collected the soil, sample, brought it to his laboratory in Sweden, isolated a bacteria. It was producing an antibiotic that was used for treatment of say cancer. And that company and that earned billions of dollars were earned by them, but African country got nothing. Yeah? Nothing. So that is over now. Now, if someone has deposited a culture, if industry are taking, although we still have a long way to go because we do a lot of hanky panky business, still we do a lot of hanky panky business so far as cultures are concerned. But otherwise, if a company is taking, say, for example, of meta-regime, they are making a formulation, they are selling it. You have deposited it, say, for example, then what are you getting? You are not getting anything, the institute is not getting anything, and they are earning. So there is a law. There is a law regarding this, and slowly, you think the things are becoming tighter. People are not understanding the things, but the government is tightening the screws by bringing what changes in the law, by bringing what changes in the way the laws are implemented. Next, this is the passport data form that has to be accompanied with the culture that has to be deposited. Let's depositor check, because we will be sending the culture and then you will have to fill it whether it is the same culture or not. If you say that no, it is not the same culture, then we will again say okay, resend the culture. This means that the culture that you have said probably it is contaminated or by mistake it got contaminated in the culture collection. Next, this is filled sheet depositor check. If you say identical or non-identical. Next, these are the catalogs. Now we are not getting printed. These are online catalogs. We are not getting them printed. Next, there is a software with which we are managing it. Next, microbial genetic resources. We have our institute website, and another is microbial genetic resources portal. If you visit it, you will get a lot of useful information regarding microorganisms. Next. Now, this is just to give you an idea of the rates that we are charging for the cultures. Identification services also, rates are different for public and private sector. Next, this is just a viewpoint of the people who have visited our culture collection. Next. No, I will not be taking much of a time here because others are also. No, I just want to tell you that we have a National Biodiversity Act. We should have a little bit aware. There is a National Biodiversity Authority. Now, as per this act, say for example, if you are going to a forest in Meghalaya, you are collecting the soil sample from there. In Here also in the protected area, you have to take a permission. In other countries, say for example, European countries, if any scientists go, if you want to visit a forest, they will, one person will be along with him all the time. If they will not allow you to take a piece of leaf or a pinch of soil, they will not allow. So here, as per this biodiversity act, if you are taking a soil sample collecting from there, so what you have to do, you have to intimate. You have to intimate the local biodiversity authority of that region. Even local biodiversity authority at the regional level, state biodiversity authority is there, district biodiversity authority is there. Then at the panchayat level also they have maintained register. They have to be informed that from such and such place it will have a location, longitude from this place I have collected a sample. So, but as of today, I think this is not being implemented in on paper also, but in the foreseeable future, things will be. So you cannot, if you cannot send any microorganism cannot be sent outside the country. If it is to be sent, permission from biodiversity authority has to be taken. Biodiversity, because sometimes we also get a request for cultures, say for example from Korea. So only hindrance is that they are directed to go to the National Biodiversity Authority, follow the procedure there. Once the Biodiversity Authority permits that you can take the culture, then and only then the culture is given. Otherwise, culture is not given. Next, 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 next. This is all about biodiversity authority. Now, another very important next, next. Now, at the same time, what we are doing is that as a utilization of microorganisms. So the scientists working on different projects, they are also trying to develop some biofertilizer formulations, or say, for example, biopesticide formulation, or any formulation uh, against any abiotic stress. Say, for example, drought. Now, archaea. Now, one of the scientists, he has come out with a formulation based on archaea. Now, in the case of wheat, say, for example, the recommendation is that 6 irrigations have to be done. If not 6, 5. If not 5, 4. If not 4, then 3. So what happens is that if you are treating the seed with that formulation, then the number of irrigations can be reduced. So it mitigates the abiotic stress. And all the formulations, it is not that the scientist had developed them, he has found very good results in the laboratory. So it is everything works well in the laboratory, but everything fails in the 99% of the cases. Microorganisms fail in the, because in the laboratory, ideal condition, in the plate everything is there. All the food is very enriched in the plate. But here in the field it fails. In 99% of the cases the micro formulations fail. So all these formulations, they are being tested through all India coordinated trials are there throughout the country, crop wise also different locations. Then the results are obtained from those. Then after that it is released. So we are working on that also there. Now another US is next, Bioinstrumentation. This is very popular. This is very popular. It is based on 3 bacteria, one for nitrogen, another for phosphorus, and third is for potassium. Now, what is happening now? The government is talking about, now what is the government saying? Doubling of farmers' income. Say, for example, when you say that doubling of farmer income, it doesn't mean that from 5 rupees it is going to become 10 rupees. Doubling of farmer income means that efforts have to be made that so that the income of the farmers increase. You all go through newspapers, you read that agriculture is at a crossroads, farmers are committing suicide. They are not getting as much as they are spending on raising a crop. And there are a number of reasons, but one of the reasons is that what is one of the most important reasons? The most, you may spend anything, you may have a hybrid seed of rice, you are taking plant protection measures, also you are irrigating, also all the operations you are performing, yet you will not get a good yield because soil health, you need that soil health has deteriorated. Sometimes we say soil is dead. The soil is, what is soil? Soil is soul of infinite life, SOIL. Means soil is anything which is living will die. Anything which is dead will not die. Okay. So now soil health means you will not find earthworms, quechua, you will not find microbes. Microbe load in the soil has decreased considerably. So insects are also there because indiscriminately use of fertilizers. So to double the farmer's income, to increase the income, what you can do? Either you reduce your expenditures, you reduce your requirement. Okay. Then what we are in, it is very popular Here also we are in Kisan Mela, through Kisan Mela. No, one Kisan Mela is going out at just now from 9 to 11, Pusa Krishi Vigyan. It is a very big farmers fair. There also our team of scientists have gone, and from people from Punjab, from Haryana, from Western Uttar Pradesh, where the farmers are, where they buy it. So what you can do is the amount of urea, the amount of SSP. Urea is cheap, then comes SSP, more costly, then DAP, more costly. Right? So you can reduce it by 20 to 25%, or say 50 to 20%. So those farmers, progressive farmers who are keeping a record of the operation, the money that they are spending, you know, the less amount of fertilizer that means you are saving money. At the same time, if you are using formulation, you are improving the soil health also. But soil health will not improve in a day. It will take years together. Just effect will not be as of urea. You spray the urea, you will, the farmer sees that it becomes green. It is slow effect. Slowly and slowly, the soil health will improve. Next, these are the different formulas. Next. Tom Curtis said that if the last blue whale choked to death on the last panda, it would be disastrous, but not the end of the world. But if we accidentally poisoned the last two species of ammonia oxidizers, that would be another matter. It could be happening now. We would not even know. No life can exist without the contribution of microorganisms. Next. Thank you very much. If you have any question, you can ask. Yeah. Our catalogs are there, but you will not be able to see the whole catalog. Okay? For example, if you need a bacillus, you will write bacillus, then all the bacillus species with their accession number will come. Fusarium, say for example, if you want fusarium, then fusarium oxysporum or Sicily, crop wise also. Then all the cultures they will come there. But if you say that your whole catalog will be open at a time, no, that will not be, because then someone will copy it. Then
there will be some bioinformation data.
We have got a bioinformatics, another facet that we different. We have got a bioinformatics screen. That is bioinformatics screen that is housed in that building.
Any question from students? नहीं, तो थोड़ा सा इनको ब्रीफ कर दीजिए, जिससे कोरोना वायरस कैसे फैला, थोड़ा सा इनको ब्रीफ कर दीजिए.
So far as the coronavirus, it originated, as you know, that at the outset I said that they are very powerful. It is a tiny. It is 1,000 time of a hair width. It is so small. Our hair as a width 1,000 part is coronavirus. But see how it has brought in. It has devastated the whole world. Now this pandemic started from China, Wuhan. Now various theories are going on that how it originated, because we all suffer from cold. These are seasonal, but you know it was it originated from there. Earlier, the people said that there was a meat market where the different animal species were there, and from there it has spread. Then this theory was also debunked. Then pangolins, they say that probably pangolins it came from pangolins. Then there was mention of bats also, from bats also it has originated. But as of today, then America was saying that the American government was saying that no, actually it was they created this virus in laboratory, but then WHO team also went there, but they couldn't prove that there was no proof of that it was artificially created in the laboratory. So as of today, we do not know. It is a RNA virus. As of today, we do not know as to how it came to infect humans. Virus, so far as RNA viruses are concerned, then viruses also have to they mutate, continuously mutate. Then the most devastating strain was Delta strain, which was very severe, which created havoc in our country. In the first pandemic, it created havoc. Now it has mutated, it has further mutated. So this is Omicron. Omicron, which has got number of mutations. I think it has got about 50 mutations. 50 mutations it has got. The thing is that we have now, the people are saying that in future also we are not to be complacent. Every precaution has to be taken, because viruses, they will mutate. They will mutate, and so precautions have to be taken in future. Whether this strain that evolves, whether that will be less virulent or more virulent. Say for example, Omicron, it is more infectious than Delta. Means it spreads very fast, but it is less virulent than Delta. And also the thing is that the vaccination, the only remedy, the only protection that we can have is vaccination. All the country. So in our majority of the population, majority of the adult population in our country has been vaccinated, and now they are thinking of giving the booster doses also. Because why happens? Sometimes the pandemic appears, it appears, then it disappears. That is why we say that the part which remains to be continuous surveillance has to be done. Continuous surveillance, which is lacking. Now, another example that I would like to give you is of I'm forgetting the name of that disease, which it was again Ebola. Ebola. Ebola means death. That means if infection takes place, 100% chances are that nobody can save. Ebola appeared in Zaire in African country, and there the hospitals were wiped out. Then crack medical commando teams came from USA. Means if a patient is there, just a terrorist has to be isolated. Immediately action has to be taken so that it doesn't fire on everybody. Crack medical commando teams were called from Center for Disease Control in Atlanta, USA. Then they control this epidemic. After that, it disappeared. Now it will again reappear, because they are. What I am trying to say is that we have a right to exist. Animals have a right. Every being has a right to exist, to remain living. Okay. In the same way, microorganisms also have every right to exist. Every right. They are also an important part of ecosystem. That is why we say, what we are going against nature. We are going against nature. Now there is a limit of going against something. Now what will happen? What is the capacity of this hall? There is a capacity of this hall. Can you push 10,000 people into this hall? What is going to happen? The same way, there is a tolerance limit of nature. That tolerance limit has been breached. We are killing everything. That is why this theory came that probably from Wuhan market, they were eating everything. So probably from there it came. AIDS also, HIV also, there was a theory that big companies they were going to be Amazon jungles, Amazon forest. They were cutting the trees. Big multinational companies. So the people there, who were hired, they probably ate monkeys. They killed a monkey, ate it, and probably through that HIV virus came to human being. So this thing is that there has to be a balance, has to be maintained. Otherwise, these microorganisms, you are seeing that in floods. In summers, there is more heat. In winters, there is warmth. What is happening? So this is going against nature. We are going against nature. The areas where apple was being grown, where snowfall, areas where snowfall takes place, there people grow apple. Now what is happening is that in those areas, snowfall is not taking place. In Kullu, for example, I am telling you, there was a valley, Kullu Valley, which was only one crop was being raised by the farmers, that was apple. Now what is happening is that in one season there is heavy snowfall, in the next 2 seasons there is no snowfall. So they have chopped down the trees and now they are growing vegetables. So this is what is happening. So balance has to be maintained.
सही कोरोना का जो वेरिएशन आ रहा है, तो मतलब क्या ये जो वैक्सीनेशन हुआ है इस पर भी अडॉप्ट कर सकता है ये?
Yes, it has. That is why the vaccine, it can overcome. Because all these questions are even the doctors, whether how much whether it can bypass it or not. Even the doctors are not able to answer these questions conclusively. But definitely, first thing is, why is a strain mutating? Why is it mutating? Because it is finding some problem. So it has to survive. It will change itself. It will change itself. The change means it will try to bypass the immune system. The antibodies which are being, it will try to bypass the immune system. So it is a evolution. It is a continuous process, and it will go on. क्या चीज? Look, I cannot answer this question. I'm not in a position to answer this question.
Sir, as we see Russia-Ukraine war, so what is the future of microbial war?
Biological one.
Yes,
The biological one.
Biological war. There is a convention against the biological war. There is a convention against the biological war. Yet, I think only a few countries, no countries might be having a cache of biological weapons. Say, for example, North Korea, because biological weapons are there. Just as nuclear weapons, nobody thinks about using nuclear weapons. In the same way, biological weapons, but maybe a few countries might be having a stockpile of biological weapons, especially this North Korea. Otherwise, nobody can even think.
यूक्रेन भी कह रहा है कि हम इसको यूज कर लेंगे.
Ukraine, it is coming in the press that probably they also have biological weapons, but I think they will think twice before using it. Because once they use biological weapons, if Ukraine uses, then the friends who are supporting them, they will turn foil. They will stop.
Thank you.
Thank you very much.
Ma'am, thanks.
यही सब बोलते हैं. On behalf of the Uday Pratap College fraternity and the entire zoology department, I would like to thank Dr. Pavan Kumar Sharma, Principal Scientist, Plant Pathology, ICAR, NDAIM MOU. Sir, you have elaborated about the NDIM. We came to know much about the NDIM and also about the glimpses of the microbial resources. So the students have been benefited, and definitely it will bring up an outcome that they have gained the knowledge related to the microbial organisms, and they came to know about various types of preservation related to the cryopreservation, subculturing, and all other identification techniques. You also talked about the National Biodiversity Act. I would like to add up that recently I had gone to Meghalaya, and I went to that double route bridge trekking there, and really it's a great thing in India. Meghalaya has a very rich biodiversity with so many different types of plants and also insects and hill stream fishes at such a great height. I could see the fishes there, and definitely it must be a very rich biodiversity of microorganisms also. I would like to add that there has to be more quarantine things related to that, because it's just like an open jungle. Anybody can take anything from there and can take it somewhere else to India, and that may it may influence the biodiversity of that place where it has been taken. So these kind of quarantine rules must be brought up, and I would suggest that you people who are working with the microorganisms take these aspects to the government so that these things get preserved more and more. You talked about the bio NPK and the doubling of farmers' income also. Yeah, so we'll come to know more about these, and I think I met him earlier also during PPT program. You had gone to the Pratap College sometime? No, that means somewhere else. I might have seen you because it looks quite familiar. And then you also talked about the coronavirus, how it is spread, and also the importance of vaccination. But then definitely we are still in much darkness about the various kinds of aspects about the coronavirus, and still there is enough of research going on. We need to upgrade our knowledge. Pavanji, and we'll have a nice session in the second and third sessions again. So thank you.
Thank you.
Point.